All right, we have a lot to do. We only have one period to do it in, and we got to get all the rest of the transcription and translation. Now, I started with the little video clip that showed everything, but it was in real time, very difficult to follow what was going on here. Now, you guys have all seen these before, all right, and it's a good thing to review because it's probably one of the hardest things to understand in biology. All right, so let's go over it, breaking it down now into smaller parts. Okay, how are proteins made? We're gonna obviously, first of all, just to remind you again what a protein is. It's the reason we're doing this now. Okay, we're gonna have a little worksheet tomorrow, 2 worksheets actually tomorrow. You'll be able to do in class to practice this because you're gonna have questions on the test. But we're doing this now because it's underneath polymers. All right, and I'm calling this. I've just created this year this new division of what I used to just have, miscellaneous organic stuff thrown together. Okay. Because I've kind of thrown in the biochemistry with the polymers. Because a lot of biochemistry is polymers, one of which, the most important of which, being proteins. All right, proteins are just long peptide chains, long amino acid chains, polypeptides. Okay. Thing is, proteins are what does everything in your cell. Proteins are why you have blue eyes, why you have brown hair. All right, they perform all the functions in your cell. For example, hemoglobin carries oxygen, insulin. All of these things that you've heard of, they're carried out by proteins. All right, now I'm going to tell you how they are made in this 2-part process. All right, the first part of that is the transcription. All right, now in transcription, as you can see in a little picture here, and you're going to see in a little video clip I'm going to show you in a minute, because these video clips now are going to be isolated on I'm going to show you 2, one on the transcription process, one on the translation process. All right, and they're going to be a little bit easier to see. Okay, although they're going to use sticks and colors and stuff like that, that's obviously not what they really look like. All right, the video clip you saw before probably is more, although you can't see it anyway, it's a little bit more accurate. But they obviously are just like different length and different color sticks on the DNA to make up the ladder. All right, but it is, it does mean something. There's a reason we're doing it that way. The first step, as you can see from this picture, is to unzip the DNA so you can read 1/2 of the chain. Okay, transcription is the process by which this information is copied. You're going to have to copy it because your blueprints are the DNA. That's what's in your cell, the blueprints. And you might wonder to yourself, well, why make a copy and send that out? Isn't that a better chance of making mistakes? Yeah, it probably is. But the reason the DNA, as we saw in that that article we read last week, why is the DNA segregated in the nucleus? What's the purpose of that? Remember, why is it in the nucleus and not like all over the cell somewhere? Do you remember? One of the defense mechanisms of the body was having it in the nucleus. So it's away from most of the toxins. It's isolated. There's less things that could damage it. Okay, making one mistake in one copy wouldn't be as big of a deal if one messenger RNA gets ruined. But if the whole DNA strand is ruined, then you're going to actually be repeating that mistake all the time. All right, so it is the process by which we're going to copy. This is what we're going to concentrate on now. First, the copying process. I don't know if the video is now or after. I think the video may be coming up now, which you have to put the.
Yeah, both. Do you record the videos too?
Yeah.
So we have balls.
Okay. Shh.
The cell transcribes the genetic designs for each protein from strands of DNA to special molecules of RNA. An enzyme unwinds a segment of DNA, making room for its complementary RNA nucleotides. Both DNA and RNA nucleotides contain a sugar, phosphate, and base. But in RNA, the sugar is ribose, and one of the 4 bases is replaced by uracil. The exposed bases of the DNA strand serve as a model for the construction of messenger, or mRNA. On other strands, the cell produces transfer, or tRNA, and ribosomal, or rRNA. In their final form, these molecules play a vital role in protein synthesis.
So that copy right there is basically the opposite of the picture. And they replaced one nucleotide base, as you're going to see. of that segment of DNA, which we call a gene. The segment of DNA that was copied is called a gene.
What was the green thing called? Is that a ribosome?
The thing that was no, ribosomes have not come into play yet. That was you're going to learn. I'm going to tell you that was basically RNA polymerase and a transcription complex, which are opening this guy up and bringing in different nucleotide bases and making a copy, the opposite copy of one side of the DNA. RNA polymerase is the name of that stuff. Okay, it basically forms a transcription complex, opens up the DNA, and brings in nucleotide bases that match. Remember, they only match in certain ways, and there are some differences, too, for the RNA versus the DNA. The transcription complex recognizes a start codon, the beginning of a sequence, and also there are stop codons. Codons are a sequence of 3 bases that tell this enzyme, which, by the way, anything that ends in ASE is an enzyme. So polymerase is an enzyme. It's a protein itself. Here's a protein involved in making proteins. That's what happens. Okay? The transcription complex recognizes the start of a gene and unwinds it. You're going to see what those start sequences look like. I'm going to have a little chart. As you saw in the little video clip, and as you can see in the little picture here, individual nucleotide bases are brought in and matched up to the other side of the DNA, and you're making a copy. You're making a single strand from these nucleotides. Okay, forming a single strand of RNA. Which, by the way, that's one of the ways. There's 3 ways which RNA differs from DNA, and that's one of them. The fact that it's only a single strand, not a double strand. There are a lot of notes to get here today in one period, and you're going to end up using them to do the worksheets tomorrow. We won't have time to do them today.
What are the worksheets
on this? There are going to be questions all on this process. So,
I can't wait.
Can't wait for that. One's kind of a neat one, though. I just found it this year. It is. Okay, so here's your RNA on this side, and here's your DNA on that side. Okay, and you can see some obvious differences here. One is obviously that one's a single strand, one's a double strand, but there are other differences too. RNA, even in the name, versus DNA. Okay, they're similar, but they are a little different. And the first difference you can see from the name, RNA has ribose for the sugar, DNA has deoxyribose. It's not a huge difference. It's just one oxygen really. It's missing. Deoxy means there's one less oxygen on the DNA than there is on the RNA.
Oh, is that what DE stands for?
DE. That's why it's called deoxyribonucleic acid. What's the other difference? You guys should remember this from Bio 1. Are they all the same nucleotides? Who's missing?
T.
Yeah, and what is it replaced with?
Uracil.
Yeah. So uracil instead of thymine. And finally, as I just said, and as it shows you in the picture, RNA is a single strand and DNA is a double strand. And you got to remember, there are actually different kinds of RNA. 3 kinds, really, but we're going to talk about, I'm going to mention 2 of them that are really going to come into play here. Messenger RNA, which you just saw being made, and transfer RNA, which you're going to see in the translation process. There's also ribosomal RNA, too. Okay? So the different types of RNA, we're going to start with the messenger one. That's the one you just saw being made. All right, and what it does is the messenger RNA carries the blueprint's instructions out of the nucleus and into the cell where it can make what it's supposed to make. Okay. So that's one kind of RNA, messenger RNA. That's the guy that's carrying the copy. But also involved in this process, you're going to have these transfer RNA molecules bringing along the amino acids. And the transfer RNA has really two major parts to it. Two parts you need to know the most, all right, about the transfer RNA are going to be this part down here, the anticodon. Guess what that's going to do? It's going to match up to the codon. The sequence of three nucleic acids on the RNA, messenger RNA, and then on the other side is going to be a particular amino acid that's going to make the protein. So the anticodon matches up, and this particular anticodon only matches for a certain number of, well, actually, it's specific to a amino acid, okay, that codon is, the anticodon is. And it will match up, and that's how it reads it, okay? So the transfer RNA contains a sequence of 3 nucleotides called an anticodon to a specific amino acid that matches that sequence. And that's how it's able to build what it needs to build. It's like an assembly line when it starts to get going, to it. You'll see.
Are we going to have one of those charts?
Yeah, I'm going to show you that chart on the next slide here. I think it's on your next paper of notes. And I'm going to ask you in a couple of different worksheets I'm going to have you do. There's a couple of different things you're going to do with those sequences. You're going to have to read them and tell me not only their sequence, what it corresponds to, but and the amino acids, as well as to their phenotypical expression based on a mythical creature. You'll see tomorrow. And I'm going to give bonus points for the person who draws the best ones.
Oh, I'm not ready to draw.
So, how are we going to draw it?
You'll see. It'll tell you how, because all you got to do is read the gene sequence.
Oh, you got to read the genes.
You got to read
I
the
have
genes.
blue ones on, guys.
Yeah, very good.
I'm not wearing
Yes. Skinny jeans.
it. Yeah, that's what you wear.
All right. So here are your possibilities. Now notice there is a possibility for more than one codon or anticodon. All right, to match up, bringing in the same amino acid. So, for example, this codon, CGU, CGC, CGA, or CGG, will all give you arginine. Okay, but some of them only have one. Methionine is only produced by AUG. Okay,
Yeah.
but others have 3 or 4 or 2. All right, and that reason you have that, there's a chance. It means that if there's a little mistake along the way, it may not cause you any problems. Okay, for example, notice all those started with CG. So you can replace it with a U, a C, or an A, or a G, and that CG is really what told it. So if there was some other mistake there, it would still give you the right amino acid. Okay, so the same amino acid may be coded for by more than one RNA codon. That helps to protect from errors in your transcription and translation process. If there is a small error, it may not cause you any problems,
Okay.
which is nice.
So somebody had to come up with this chart. They had to think about all that.
Well, they didn't think of it. You actually configured. We've mapped the DNA. We've mapped the human genome. We basically, we can predict. There's a lot of genes that have been isolated. We know this is the gene that produces insulin, or this is the gene that produces whatever. All right, there's a lot of genes like that we know what they do. So this isn't just made up stuff. We know that that's what they code for.
They still can't make people yet.
Yeah, that's a little harder. Right? Yeah, a little harder to make people. It's it's one of those things. It's unfortunate that a lot of scientists do. You have to going all the way back to Frankenstein. I mean, you have the people. Mary Shelley. The book. You have people who always think that they, by being able to explain something, it means you actually can do it. It's not the same thing. Like for example, something really good. Just to give you an example, sports is what usually you guys know the most about. So a heck of a lot of sports writers out there. You ever look at them? You ever watch like Pardon the Interruption or those other shows? These guys never played a down in their lives. They can talk about it pretty well. They can understand the rules, but they don't know. They never played the sport. All right, and the same thing's true. I mean, I can tell you what these. It doesn't mean I can do it. It's just like I can tell you how LeBron James scored this many points last night. I can give you every stat about him if I'm really an aficionado of basketball. But it doesn't mean I can play like LeBron James. All right, it's a big difference.
Did you see that fight?
I could.
Did you see that fight?
Some codons that basically tell the sequence when to start and stop. So when it reaches a stop codon, like these here, all right, it will just know that that's it. You're going to stop reading. You're going to stop bringing in more amino acids. You are done.
What happens if it's a stop and then happens if it's another stop codon?
To get into an argument, you start rolling.
They argue a lot.
That's not like
I didn't know if that
mutation.
ever happened.
Yeah.
Like,
It's not a mutation when.
No. The stop codon is supposed. You're mistaken. You see, you're making a chain of 120 protein or 120 amino acids to make this protein. At the beginning's a start codon, and at the end of that 120 sequence, after it's got all of them in there, there'll be a stop codon. It's supposed to be there. It's not a mistake. It's supposed to be
what
there.
happens if there's a stop codon halfway through?
Well, then that would be a mutation.
That's right.
That would be a mistake, and it would probably make nonsense. That would just be useless and wouldn't make that protein. But that doesn't. If that happened, for example, in the transcription process, no big deal. You made a useless protein, but as long as the DNA was unaffected, the next time it copies it, it'll copy it correctly, hopefully, and you'll make the actual thing correctly. All right. So the stop codon is supposed to be there. If you did have an extra one put in by accident, that would make a useless protein most likely, but it's okay. That's why, remember that article we read, the cancer article? You want to protect the DNA. If the mistakes in the messenger RNA, say you made a crappy protein one time that can't be used. If the mistakes in the DNA, you're going to continue to make the crappy protein, okay?
That's how you get.
Right, okay. Translation. Let's do a little video of this in a second after this introduction. Translation is where you take the ribosome and the transfer RNA, bring together onto the messenger RNA the individual amino acids. Okay, you translate, you turn that information into an actual product. It's like the assembly line. If DNA is like the blueprints, the ribosome is like a factory. And the messenger RNA and the whole process is like an assembly line in that factory, where you take the actual information from the DNA, you run it through the messenger RNA, the ribosome, and with the help of the workers, the transfer RNA, bringing in the individual parts of the molecule you're trying to make, you, in an assembly line, assemble the actual protein. Remember, a protein is just a series of peptides, a series of amino acids, polypeptide. Okay. First, the messenger RNA travels to a ribosome, and it consists of two subunits. With the video you're going to see is going to say there are 2 binding sites. All right, it's just it doesn't really matter how many. I'm not going to ask you about the P site or the A site. I'm not going to go into that kind of detail. All right. The key is the messenger RNA travels to a ribosome, which does have 2 basic parts to it, and that is run like a ticker tape through it, reading every 3 nucleic acids as a codon, and then bringing in a specific protein or specific amino acid that matches that guy up. All right. So let me show you a little video clip here.
Translation is the process by which the information contained in messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has two binding sites for transfer RNA, the P and A sites. Transfer RNA carries an amino acid, which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies that particular amino acid. A ribosome assembles on the start codon AUG. Transfer RNA with the anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an anticodon complementary to the second messenger RNA triplet. The amino acid carried by the second transfer RNA binds to the methionine. The first transfer RNA leaves the P site, and the second transfer RNA moves there, still bound to messenger RNA. This brings the third messenger RNA codon to the now empty A site,
So it's just like
and
an
the
assembly
appropriate
line.
transfer RNA can bind to it. The third amino acid is added to the chain, and translocation occurs once again. This process of polypeptide chain elongation continues until a stop codon is reached. A release factor binds to the A site. It carries no amino acid, but facilitates release of the polypeptide and the messenger RNA from the ribosome.
It just randomly fall.
After a stop codon. Now, that was just the process of translation. All right. Now, I obviously you can't watch the video every time, but I do have these pictures that are all on that last paper here that kind of illustrate what's going on here. Okay. And it is just like an assembly line. All right. You're trying to make your product is the protein you're trying to make, the polypeptide, and you put them in one at a time. All right, and they match up. And like I said, there can be mistakes, and if they happen at this point, it's not as big of a deal as if it happened by damaging the actual DNA. Now, the complementary tRNA molecule, it comes and binds to the codon. That's at one of the two sites in the ribosome. It's now the amino acids, physically are right next to each other, and they will connect. You saw how that happens, all right, in the past. It's like an amino acid. One's a base, one side's a base, one side's an acid. You put them together, they're going to react. So they get really close together, and that's what happens. You link these guys together. Once that bond forms, okay, once the bond formed and you lose the amino acid from one of the transfer RNAs, he breaks off and it slides down. The other one slides down and gets ready for the next guy to come in.
So the ribosome is only in the
Yes, translation part, right. And where are ribosomes located? Remember what structure in the cell, one of the organelles in the cells that they were located on? You remember? What was the name? A big long name.
Oh crap.
It begins with an E.
Endoplasmic.
Yeah.
Endoplasmic reticulum. Yeah.
Is there a rough?
Rough ER is the stuff that has the ribosomes on it. Right.
That's a rough ER.
That is.
People die all the time.
Now, if I know the actual codons down here, I know which amino acids are going to be linked together. That's going to be one of the questions I'm going to ask you in the worksheets, which we'll probably start today because we'll probably have a few minutes. Your class did not finish them, so you might want to start them today. You won't finish them today, but you might want to start them today because tomorrow a full period won't be enough time. we will be getting the notes tomorrow for the real polymers. The good lab, we're going to be doing one that you've been waiting for, the one that has we make rubber balls, slime, and all that stuff.
This test is going to be on.
This test is going to be on biochemistry and polymers only. There's a lot of, I said to your class, there's some other biochemistry I'm going to have in here. We're going to, remember the soap lab we did?
Yeah.
Well, we're going to test the bactericidal action of soap. We're going to try to grow bacteria, we will grow bacteria, and see what our soap does, and other things too.
Oh, I thought I was going to be able to use the soap.
You will. You only need to scrape off a little bit to make a solution. And you should have plenty to do that. So we have that lab in here. We have, so that's going to be a little bit of notes on bacteria we're going to talk a little bit about. I'm going to do a little bit on lipids, and I'm going to do a little bit, of course, we have a little bit more to do on actual regular polymers. All right, so we won't have this test, I think, until probably next Tuesday, maybe. All right, not this week, but next Tuesday. It's possible to be, no, it won't be this week. It'll be next Tuesday. All right, last two sentences here to write down in these notes here. What happens after that? It continues. It just goes on and on and on and on, hundreds of times until you've made your amino acid chain, the polypeptides, which is the actual protein, which gives you the hair color you have, the eye color you have.
Is it true that people with brown eyes actually have blue eyes, but it's just underneath like a layer of
There's,
something?
I don't know. I think it's like leaves. Certain pigments mask other ones in the fall. As the pigments, as the chlorophyll in the leaves dies, you start to see other colors that are already there, but they're just masked. So I suppose if you had no pigment at all, your eyes would be, I suppose they'd be blue or white,
Ooh, that'd be
clear,
weird.
like an albino.
Tommy Lama.
He's not an albino
How do you know?
because he's not from Albania.
Hey, but Sean, you know how you make holy water?
No.
You boil the hell out of it.
Good.
How do you make a tissue dance?
That I don't want to hear this one. I have a feeling.
You could put a little boogie in it.
Okay, last one. Polypeptides. Eventually, by the way, these things they're not quite ready, even when they are, even when they're all hooked together. They end up having to fold upon itself, and there are different other molecules in the cell that do this. It's not important we talk about that, but they do get folded up on themselves before they finally are usable. And that usable thing is pretty much everything in your body. Every enzyme, every protein, hemoglobin, insulin, the ones that you're familiar with, pretty much everything your body does is done because of proteins. Now I've got one little summary thing, very dramatic, could have been made by Disney, to show you all these again from soup to nuts.
But you don't know boogie in it.
With no boogie. So from the beginning to end, I have a little video clip like we had in the beginning, well, not the same one though, which showed you the whole process. This is kind of like a dramatization. It's got a couple of really good, now that you've seen the whole thing and got notes on it, it's got a couple of really good 3D molecule animations toward when we get to the translation process in particular, I think. So you want to pay good attention. It's not just little sticks, is what I'm saying, with a V or a U or a triangle sticking out of it. It's kind of neat. And it's only a couple minutes long. And so hit that like for me again.
Did Disney make a new Star Wars movie?
I heard they were making one, but they blocked it. Okay, shh. Very dramatic.
With computer animation, we can enter the cell to view this remarkable system at work.
Those are nuclear pores, remember. And that's what?
After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA.
and it has to leave.
When
the
transcription
nucleus.
is complete, the slender RNA strand
And that's why
carries the
those pores
genetic
are there, to make
information
sure
through the
nothing gets in or
nuclear
out that stops the
complex,
host there.
the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins.
That's pretty. I like this. The best animation really does show you everything very well.
Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
Okay. All right, get the lights for me. That's it.
It
It was pretty intense. I think you should get a.